The term cloning describes a number of different processes that can be used to produce genetically identical copies of a biological entity. The copied material, which has the same genetic makeup as the original, is referred to as a clone. Researchers have cloned a wide range of biological materials, including genes, cells, tissues, and even entire organisms, such as sheep.
Many plant species and some animal species have natural methods of cloning. Many species can reproduce asexually and hence possess natural methods of cloning. All bacteria, the majority of fungi, and many species of protists reproduce asexually to produce genetic clones. While most plants reproduce sexually, they also possess methods of asexual reproduction. Certain animal species can also reproduce asexually via a variety of different mechanisms. The natural plant cloning method is another natural process of cloning. Plants have the capacity for vegetative propagation, whereby small pieces can be induced to grow independently. This is because adult plants possess meristematic tissue capable of cellular differentiation. Virtually all types of roots and shoots are capable of vegetative propagation. Garlic and onion bulbs are modified plant leaves. All the bulbs in a group are genetically identical. Underground stems, such as potato tubers, can form new plants which are genetically identical to the parent plant. Certain plants can form horizontal stems called runners or stolons that grow roots and develop into clones. Some plants can reproduce asexually by producing spores. Spores are also produced by certain types of bacteria and fungi. The natural human cloning method is another one. Even human beings are capable of creating genetic clones through natural means. Identical twins are created when a fertilized egg splits in two identical cells, each forming an embryo. Non-identical twins are created when an unfertilized egg splits into two cells and each is fertilized by a different sperm. Identical twins will be clones of one another, while non-identical twins will share 50% of the same DNA. As far as artificial cloning, animals can be cloned at the embryo stage by breaking up the embryo into more than one group of cells. At a very early stage, embryonic cells retain pluripotency. These cells will differentiate to form all the different tissues comprising the organism. If these embryonic cells are separated artificially in the laboratory, each group of cells will form cloned organisms. This separation of embryonic cells can also occur naturally to give rise to identical twins. The separation of embryonic cells has to happen early in the development cycle, ideally around the 8th cell stage. The separated groups of cells are then implanted into the uterus of a surrogate to develop into genetically identical clones. This method of cloning is limited by the fact that the embryo used is still formed randomly via sexual reproduction, and so the specific genetic features of the resulting clones have yet to be determined. A second and more reliable method of artificial cloning involves somatic cell nuclear transfer. This involves replacing the haploid nucleus of an unfertilized egg with a diploid nucleus from an adult donor. The advantage of this technique is that it is known what traits the clones will develop. Reproductive and therapeutic cloning is another method of artificial cloning. This method of using differentiated cells to generate cloned embryos can be used for two main purposes. The first one is reproductive cloning. If the embryo is implanted into the uterus of a surrogate, a new cloned organism will develop. The other one is therapeutic cloning, where the embryonic cells can be induced to differentiate to create specific tissues or organisms for transplantation.
Cellular cloning is the process of producing a group of cells that are genetically identical to a single ancestor clone. Basically, it is the making of another duplicate of a biological material. Vegetative cloning is the process of increasing the number of plants of a particular species or cultivar. This allows plants to be created in ways that nature cannot duplicate. As you all can see from the picture, it is a visual representation of what vegetative cloning would look like. There are two methods of vegetative cloning, one being asexual and the one being bisexual. An asexual reproduction would be producing a new plant by an original part of the plant, like the leaves, stems, or roots. Now, there are some disadvantages of doing this because sometimes there could be a plant being used as a clone and may have a disease. Therefore, it could potentially harm the other plants and all crops being cloned by the infected plant, and it could all be lost. Embryonic cloning is the process of using a human embryo cell to reproduce or create a copy of an embryo. This topic brings about many controversial political discussion because many people have different views of an embryo. There are people who are against embryonic cloning because they believe that an embryo is already a human and destroying its life for an experiment is against their beliefs. Others are for embryonic cloning because they believe that it could have the capacity to potentially bring upon a solution for diseases that have not been found any treatment for. Dolly, born in 1996, was cloned from an adult sheep. She became very famous and well known due to her cloning status, for the reason that she was the first animal to be cloned with an adult cell. Dolly was cloned by Keith Campbell and Ian Wilmot. Many people were fascinated that she was cloned from the cell of an adult mammal. Dolly was cloned by scientists by getting an udder cell from an adult sheep and injecting it to an unfertilized egg. Snoopy is the first dog to be cloned from the cell donor of an Afghan hound named Tide. Originally, there were 2 dogs being cloned, but one died a few weeks after being born. Therefore, only Snoopy became the first successful cloned dog. Snoopy's invention was considered to be the most amazing thing that ever happened in 2005, according to an article published by SNU. Cloning dogs is not very efficient as we found that many embryos were used and only one embryo was able to be cloned. Scientists took the skin cell from Tide, the Afghan hound, and then inserted the DNA into a dog's egg. In addition to, Snoopy was seen as an experiment to scientists.
Somatic cell nuclear transfer is also simply known as nuclear transfer. This is different than artificial embryo twinning, but it still produces the same result, which is a clone. This was the same method used to create Dolly the sheep. So a brief biology lesson. A somatic cell is any cell in the body other than reproductive cells. Reproductive cells are also known as germ cells. In mammals, every somatic cell has 2 complete sets of chromosomes, whereas the germ cells have only 1 complete set. The nucleus is a compartment that holds the cell's DNA. The DNA is divided into packages called chromosomes, and it contains all the information needed to form an organism. The transfer process involves taking a somatic cell from an organism. Then, you remove the nucleus and all of its DNA from an egg cell. Next, you transfer the nucleus from the somatic cell to the egg cell. The egg cell, with its new nucleus, starts to behave just like a freshly fertilized egg. It is then developed into an embryo, which is then implanted into a surrogate and carried to term.
Although controversial, there are some potential benefits to animal cloning. The first one is that it can produce the best animal population. Cloning can be a means to create an animal with identical genetics. This is a good thing because all animals will have the same healthy genes. The second benefit is that it will reproduce endangered or even extinct species. The DNA of extinct animals can be cloned to reproduce them. Endangered species will no longer be in danger of going extinct because cloning can produce more of them. As a result, future generations will be able to see those animals. Lastly, it can be a way to quickly create more sources of food.
Some possible medical benefits of cloning humans could be defective genes could be eliminated. As humans continue to reproduce, damage to their DNA lines increase. This creates mutations and defective genes, but these could be eliminated thanks to the cloning of healthy human cells. Second, faster recovery from traumatic injury. Recovery times could be decreased and true healing could occur thanks to the cloning of their own cells to help the recovery process. Third, infertility could be eliminated. This would create the opportunity for an infertile couple to experience the joys of having a family without enduring the painful infertility procedures that are common today. Cloning dangers that have been seen in animals. Often the problems include severe developmental delays, heart defects, lung problems, and malfunctioning immune systems. The cloning process seems to create random errors in the expression of individual genes. Those errors can produce any number of unpredictable problems at any time in life. There have been multiple failed attempts at cloning. Dolly was the only clone to be born live out of a total of 277 cloned embryos. Clones are also subject to a shorter lifespan due to the age of the cells issued in the cloning process. The health effects that cloning has had on animals is a cause for concern for cloning.
Now you may be asking yourselves, are there any dangers for human cloning? Well, human cloning has yet to be done, so we do not know what the actual effects would be. But from animal trials, we can assume the type of dangers that could be possible halts further thinking of human cloning.